Well, thank you all for supporting and joining this MHRC seminar. Now, it should be chaired by our interim director, Professor Sylvia Chen, but she's too humble and asked me to do it. And now I think I should be the formal interim director. But I was too busy, and that is why I asked for the senior management that I stepped down a few months ago. But for this speaker, although I have stepped down, I still would like to chair this very important, and I would say would be very inspirational seminar on the topic, and that is very interesting. And I will be introducing our speaker later on. Now, just a little bit of background is that how I get connected to Professor Liang. Everybody knows that in Hong Kong, we have a scheme called STEM professorship. So it was dated back to 2 years ago, and then I was still head of the Department of Rehabilitation Sciences. And I have to look for, potential candidates around the world, especially from very high-ranking university like, Harvard, Stanford, MIT, and so on and so forth. And I sent an email to LIANG, and he did not answer me until 1 year later. It has been a very long wait, all right? But I'm still waiting for his reply. And after 1 year, and he replied, and all of a sudden, when I woke up in a very typical morning, I checked my email, and I saw, Professor Liang's email, and asking me whether I'm still interested in him. And I said, of course, I'm always interested in you, in, exploring, further collaboration with our department, with MHRC, and also with PolyU. And then he said, I'm now more ready to explore. And that is why we have arranged this short-term visit. And I hope that, with this short-term visit, and he will be visiting us more long-term, and eventually for some, long-term collaboration. I think this is, the things that we have been hoping for. Am I correct, Sylvia? All right, okay. Now, because, our Professor Liang is the first time visiting PolyU, and also MHRC. So I just follow the routine that as the chair, I just would like to summarize a very basic biographical sketch of our speaker and also his major achievement. All right, so bear with me. Now, Professor Li-Ming Liang is currently Professor of Statistical Genetics at the Department of Epidemiology and Biostatistics of the T.H. Chan School of Public Health in Harvard University. And he completed his education with a PhD in biostatistics from the University of Michigan. And after that, Professor Liang joined the Harvard Chan School as assistant professor in 2009 with a secondary appointment in the Department of Biostatistics, and was promoted to associate professor in 2015 and then promoted to professor in 2023. And Professor Liang's research focuses on the development of computational tools for multi-omics data analysis in order to enhance our understanding of the biological mechanisms of diseases and to provide a predictive model for the risk and benefit assessment of an intervention. He actively applies his methods to large-scale studies, which include unique longitudinal cohorts with a risk for asthma, allergy, lung cancer, COPD, and more. And Professor Liang is a very productive researcher. He's the PI of 5 current and incoming research grants and co-PI of 9 grants funded by the NIH or other national institute in the States. And he focuses on obesity and also chronic diseases. In total, he has secured more than $22 million for his research grants. And Professor Liang is highly prolific. He has already published about 30 research outputs focusing from a wide range of important topics related to chronic diseases. His works are published in Nature, Nature Genetics, Nature Review Genetics, Nature Methods, Nature Protocol, and Journal of Allergy and Clinical Immunology. Today, we are very happy that he's now here physically, and he would like to share with us a topic on computational pipeline for cross-trait analysis with examples for psychological and neurological diseases. Let's welcome Professor Liang with a round of applause.
First, thanks Hector for this such nice and warm introduction. And thanks very much for inviting me to share our research with the group here. As Hector mentioned, we are looking for some long-term collaborations and to any degree that everybody are happy with. So I'm very happy today to share what we have been doing in the past several years. So when Hector invited me and we talked about the two talks. One is the general open talk to the public. The other is a talk for internal talk. So I tailor the one for internal talk to be more psychiatric or neuron degeneration, neuron disease. But later on, I was asked to just give one talk. So I think this might be the best topic that for the group here, because everybody seems to be very interested in psychiatric or neuron disorders. So today I'm going to share the two series of research that we have been using based on cohorts and large epidemiology study with multi-omic data sets. And I hope to use this as example to sparkling idea for collaborations and new research. As you can see, my research mostly focus on human large cohort study, longitudinal follow-up research, but we are very keen to collaborate with basic scientists to understand what is the function and the biology behind the genes, the protein, the metabolite we identify, and how eventually to translate this into real application like intervention or drug development. Right. So I would like to give a bit more background about the motivation of these kind of studies. So right now you can see in terms of the advancement in biotechnology, we can easily measure all kinds of omic data like genomic, metabolomic, proteinomic, and gut microbiome in a large set of individuals, several thousand, even tens of thousands. Like UK Biobank, you have several hundred thousands of individuals in this cohorts. So this gives us a very good opportunity in terms of objective way to measure the exposure and the risk for disease. In traditional epidemiology study, like a nutrition study, you usually ask people, what do you eat in the past week or in the past months? But those kind of questionnaire-based assessment are subject to bias, subject to measurement errors, not reflecting a lot of biology of what they eat and also what they respond. Because even we eat the same thing, our body might respond differently because of different genetic backgrounds, right? So now it gives us the opportunity to integrate this multi-omic data set in large-scale longitudinal cohort in terms of two things. One is to understand the mechanism of the disease. What's happening when people get in the disease? And the other is to predict what is the risk in the future for having a disease and what is the efficacy of treatment, and also what is the exposure, like dietary exposure. Can you have a more unbiased and accurate measurement for dietary exposure compared to the traditional questionnaire-based assessments? So this is the overarching goal in my lab in the past several years and also in the next immediate futures, is to how to using molecule profile to characterize individual in terms of their risk and understand the mechanism of the disease and eventually have some translation opportunities, right? So I like to highlight several important points to how to integrate multi-omics data in the hypothesis-driven studies. So first, we should understand what the omic brings to us. First is give us the new territory for biology, because so-called omic, we are measuring all kinds of things in the similar catalog, like all the protein, like 3,000 or 5,000 protein, all at once, not just the candidate set of proteins. Like genome, you get the genetic variant and the whole genome, right? So you don't miss any of the others that you don't show up in a hypothesis or don't show up in the literature, give you a hint, but you can have the opportunity to discover these novel biologies. And also many of these effect size could be small, but once you have the data available, by combining them together, you can get a lot of power in terms of prediction or assessment. So this has been seen in polygenic risk prediction, like genetic risk. When you add up all the small effects for genetic variant, you can eventually give you a much better power for prediction and more accurate, right, compared to the top genes or top genetic variant identified from genome-wide association studies. So by integrating this analysis, we can understand what is the functional cause and what's the downstream or upstream effect for exposure of interest. And also we can link between disease and trace that they're sharing common genetic effect. So this is called the pleiotropy or causal link between them. This is important because in traditional epidemiology study, we are also worried about the causality, right? Whether association means causal. This is the key questions. But in genetics, if you find the shared genetic, there must be some intrinsic association between the two disease or traits, because there's no reverse causation here. Having a disease do not change your genes, right? We are talking about germline mutation. But having the gene and then having a high risk for disease, this is just a one-way effect. So by leveraging this effect direction only from the genetic variant to the disease, then we can have a reliable estimate for the association without subject to environmental confounding. Right. So because disease are heterogeneous, like asthma can be lung allergy asthma, and you can see Parkinson's disease can be many kind of depression or psychiatric or schizophrenia, for example, can have a multiple different biology driving the disease. So by leveraging the sharing of genetic between different disease or traits, you can understand better what is the subpathway that drives different subtype of the disease. So this is another advantage by integrating multi-omic together. And finally, we can have a more objective way to measure the exposure compared to the traditional questionnaire-based assessments. So when in terms to formulate the hypothesis, because we don't just put all the data together and hope that the analysis can just put up some interesting findings. We need to have a very careful design, or what's the scientific question that you think that's important. So I want to lay out this way for hypothesize definition. For example, we treat the genetics here and the environmental exposure to be out of the body. Even genetic is inside the body, but when you have the genetic variant, you have the whole life. Right? It doesn't change the germline mutation. But for genetic, for gene expression, epigenetics, metabolomic, proteomic, this keep change all the times, right? From the morning to the afternoon, and from your life to when your childhood or adults. So this could change, and eventually, it's the phenotypes. We can interesting a lot of different kind of phenotypes. So now it becomes an omic, so phenomic. So how do you, based on your scientific interest, how do you formulate the hypothesis based on the diagram? I think it's important. So then I also want to bring to your attention about the time scales of the life course of this omic. So this is for somebody the whole life or a person. For genetic, it represents the whole life, the lifetime burdens of the genetic effect, right? Because you have this gene, then you have this all the times. But for epigenetic, it's related short term. Say, for example, DNA methylation, it can be quite stable for a few years, but it can change over time. For metabolomic, it's even shorter time, like 48 hours. After 48 hours, it metabolizes, metabolism being finished, and then some metabolite would go away. But if you eat the food again, it comes up in your body. If you continue consistently eat same food, so it will consistently occur in your body. So it can give you a longer term impact. So I want to bring this into consideration when you integrate the multi-omic data together. Right. Now you place the timing for screening and intervention, right? Early screening time, early intervention, and then what's the time for disease occurring and the treatment efficacy to be measured at a later time point. So I want to build a time scale in your hypothesis, in your study, not just for the biology, there's a one snapshot, but also the times. Because the disease needs time to be developed. The disease would also have time to be cured and prevent. Right. So for example, in the genetic part, I'm going to show the cross-trait genetic between asthma and mental health disorder, including depression, anxiety, and ADHD. I'm also going to show the Alzheimer's disease and cancer and metabolite traits shared genetics, also between resting heart rate and diabetes. I handpicked this for the trait that relate to neuron disorders. For methylation, we have been looking at DNA methylation and PTSD, but I don't have time to present this study today, but I just want to show you that we also have this study ongoing. For the metabolite study, I'm going to show the MIND9 interventions and the effect on the brain expression and eventually to dementia and cognitive functions outcomes. So I'm going to show it later, last example today. So back to the genetics. we know that there's a natural direction effect from the genes to the disease. There's no reverse. So having disease do not change your germline mutation. So this gives a good opportunity to integrate multi-omic to the causal diagram. And then because there's a larger scale GWAS with the biobank and consortium published summary statistics, give us the more precise estimate for the genetic effect. So this is a good time to leverage this effect in your longitudinal studies. So, and also this study, the result can be shared across studies. You don't need to worry about privacy issues and sharing data issues, so become more public available. So we can use this. So, all right. So I want to give an example using the asthma as example. The motivation behind this is because we recognize asthma is a highly heterogeneous disease. There's allergy asthma, non-allergy asthma. They are sharing some biology between different subtypes of asthma. They also have distinct biology among these different subtypes. So how do we understand asthma better? By leveraging the cross trait between asthma as a primary trait of interest and the shared genetic between asthma and allergy status, allergy disease like hay fever, rheumatoid arthritis. So, for example, we published in Nature Genetics 2018. It's looking at how this allergy condition in asthma. So trying to understand what's the allergy asthma perspective or shared pathway. But also we know that for non-allergy asthma, obesity is also a high risk factor for asthma. So we look at how BMI and obese status can share with asthma in terms of the genetic effects. So this is another study to identify maybe the different pathway than as allergy asthma, right? But we also noticed that some mental health disorder also have some phenotypic association with asthma. So we're trying to understand the biology behind this. So we try to look at the shared genetics between mental health disorder and asthma. So we published in European respiratory journals in later years. So you can see by looking at asthma as a primary trait and the multiple different kind of trait as a secondary trait, you can understand the subtype or subbiology pathway related to asthma and different subtypes. So I think this strategy will be quite useful to extend to other disease like neuron disorder, as long as you can select one of the disease as your primary trait of interest, and then carefully select the secondary traits that you can readily apply these methods. So also we can try to use the genetic effect to distinct this causal link. For example, for anxiety and asthma, we do find the shared genetics, but when we put in the causal inference, we don't see there's a causal link between anxiety and asthma. So neither asthma causes anxiety or anxiety causes asthma, but they do share genetics. Maybe they have shared common genes, but they don't cause each other. But on the other hand, obesity like BMI has a very consistent effect to asthma, not significant effect from asthma to BMI. So this is the quite imbalanced effect. So from obesity to asthma, it's quite clear. And the causal inference are pretty significant. So with this strategy, we apply to a range of disease. You can see from Alzheimer and cancer, the metabolic trait that we talk about today. And we even look at COVID, severity of COVID and non-allergy asthma has a shared genetics and obesities and also lung functions and based on the China category biobank data sets. So we find this strategy to be quite useful. So we we summarize this strategy into a computational pipeline. So that's why the topic, title of my talk. We outline the several phases of carry out this strategy by starting from curating the GWAS summary statistic to the whole genome sharing genetic effect in the phase 2. And then followed up by individual analysis for cross-trait meta-analysis to identify individual genetic variant sharing between the disease and linking to functional annotations like leveraging the gene expression tissue data in the GTEx portals, and also the cross-trait meta-analysis and also the causal inference relating the different traits. Right. So we find this is a good strategy, even though we are using asthma as an example to illustrate this method, but I think this is really helpful for other disease. And we also talk about the different pros and cons for different methods using, for example, to estimate genetic correlation. There are multiple methods. We talk about the advantage and disadvantage. We outline this in the paper. I'm not going to go through all the details here today, but I want to give you that there's a reference here that you can look at the detail, especially for Mendelian randomization analysis. to estimate the causal effect between the traits. There's quite a bit different assumption underlying different methods. We should be very careful about how to use different methods and how to interpret the results of this. Okay. Then I'm going to go through several examples just to motivate this, how this we find from this strategy. Looking at asthma and mental health disorder, there are background that to show in epidemiology study that asthma, anxiety, depression, and attention deficiency or disorder has similar link between in the observational studies. And there are also clinical evidence to show some psychosocial factors affect asthma pathology in both children and adults. And also, they also find some specific link like ADHD and asthma or depression and high neuroticism, but not anxiety in several studies. So this motivates us to look at the shared genetic component between asthma and different disease. So we start with curating the data from published ADHD, anxiety. This is the eating disorder. This is PTSD. And then autism spectrum disorder, bipolar, and depression, and schizophrenia, a range of psychiatric neuron disorders. And then we try to estimate in the second stage, what is the overall genetic sharing between the disease, overall, whole genome by whole genome. If they do show overall shared genetic, we go to the next steps to look at how the individuals are sharing at individual variants or functional causal set of variants. And then what is the function, like what is the gene expression in what tissue are affecting by the genetic variant share between asthma and these disorders. So looking at this by GTEx data set and colocalization analysis. And finally, we use the Mendelian randomization analysis to try to teasing out the causal effect between them. So as you can see, this is a strategy we apply to asthma and mental health disorders. And if you take a look at the overall shared genetic at the whole genome scale, you can see that quite substantial shared genetics. These are the p-values. For example, ADHD and anxiety. And also here is multi-depression, major depression, depressive disorders have a substantial magnitude of shared genetics. And also their statistical evidence are pretty strong. The p-value are pretty small. So for these three, so we will follow up into the next step is to look at how individual variants, like showing up here, the most significant genetic variant, and the regions nearby the SNPs to be between asthma and ADHD and anxieties. And this is for depression, major depressive disorders, and many more genetic variants identified. So you can see that it's down to the genetic levels that they do share these genetic effects. Now, next, we try to leverage the gene expression data set that has been computed in prior study. This is based on the genotype tissue expression project in the NIH roadmap project to learn what's the genetic effect for all the gene expression in all different kind of tissues. By estimating the genetic effect ahead of time, you can try to impute this genetic effect of this expression into your own study and then link those gene expression with the disease of interest. Right? So basically, this is time to look at the shared genetic effect between the gene expression in a specific tissue and the disease of interest, like a trait here. Of course, it can be subject to different hypotheses. For example, the gene has a causal effect to the disease, or there's a reverse causation, like disease causing the gene expression, right? Now, because this is expression, it can have a reverse causation, right? Unlike the SNPs, it cannot be reversely caused by the disease. Gene expression can be reversely caused by the disease. Or the gene expression can have a the pleiotropy effect with the disease, but they don't have the causal effect between each other. So this analysis can pick up the signal of any kind of the three here. But in any of them, they do share the genetic effect. So we should be very careful about interpreting the finding of this. And this is a good resource. If you don't know the GTEx consortium, then this is a very good resource to explore later. This is based on about 900 donors. They donate from 30 to 40 tissues and then measure gene expression and the whole genome genetic variants in different kind of tissue. You can see different sample size in different tissues I think this is up to the release, the release 7 or release 8. And then they learn how the genetic effect for different tissues expressions. With this information, we can try to estimate the shared genetic effect between expression and the traits. Okay. So for example, looking at one specific gene like ORMDL3 and at the chromosome 17, which is the asthma SNPs and the genes that we identify for asthma, you can see their genetic effect across different tissues. Some are pretty significant, like the cells and EBVs and spleen and whole blood are pretty strong genetic regulation on expression in those tissues. Some are even has the different effect directions. So you can see even the same genetic variant and the same gene, they can have opposite effect regulation in different tissues. So we should be careful about this. Okay, so the top one here is the liver and the breast tissues and adipose tissues here. And then the downstream here is the blood and EBVs and artery and other tissues. Okay, so this is a good resource to integrate with the disease genetics. So with this, we can identify for what genes and what tissue are shared with asthma and shared with mental health disorder, different kind of mental health disorder like ADHD and MDDs. And so you can have another level of sharing, not just the genetic variant, but also the gene expression and specific tissues. So you can understand the biology more. So at this point, I would be very interested to collaborate with our basic scientists to understand what is really driving this sharing, what's the really function of the gene at that tissues are responsible for asthma risk or the mental health disorders. So next is the analysis for so-called the Mendelian randomization analysis. Not sure if you are familiar. If you are not familiar, you can see that this is the analysis trying to understand the potential causal link between exposure X and the outcome Y. But it heavily based on several assumptions. One is that you need to find a genetic variant that's strongly associated with the exposure X. This can be easier to find given the larger scale genome-wide association study, and the published available study is available for you because the larger scale study, you can identify strong genetic instrument for the exposure X. And next, there's no confounding factors between the genetic variant to the outcome. This is also usually acceptable because in the GWAS, you control this confounding by principal component of the genetic variants. Therefore, arguably, population ancestry is the only confounding factor for GWAS, but this can be easily handled in the larger scale GWAS. This is a standard analysis, right? So this is okay, but the last one is very hard, right? We assume that the genetic variants affect the disease, like PCSK9 affects CVD only through LDL, cannot be through other pathways. Right? Only if this is the case, then you can estimate the causal effect by simply divide the total effect to the effect from genetic variant to exposure to get the causal effect from the exposure to the Y outcomes. Right? So as you can see, this is not easy to satisfy assumptions. So many different kind of Mendelian randomization analysis methods are trying to relax this assumption from different angle, and then try to tackle this in a more general settings. So usually we recommend to apply several different methods to see if the causal effect are consistently out from different methods. So it will be more robust to different assumption for this is the third assumptions. Okay, so with this analysis, we try to understand whether asthma and ADHD, asthma and anxiety and MDD are caused each other. So as you can see, for example, ADHD, from ADHD to asthma, this direction seems to be more significant comparing to from asthma, the reverse direction. Okay, so having ADHD first and then asthma later on in their childhood, and maybe adulthood asthma as well, because here we don't distinct childhood and adult asthma. But for anxiety, seems neither direction seems to be significant. So it seems to be consistent with the shared genetic, but no causal link between the two. For MDD, it seems to be also the forward direction from depression, depressive disorder to asthma later on. Okay. So yeah, this is the conclusion that we seen in the data. But I would like to follow up with this study with more larger scale study as well as more functional studies to understand the shared genes and to try to understand the mechanisms as well. Okay, so that's the first example. The second example is looking at resting heart rate and diabetes. I put this here because resting heart rate is reflecting a balance of the sympathetic and parasympathetic input, controlling by the neural biological network and hormone system as well. So the literature has been very interested in how resting heart rate are linking to diabetes. They're correlated, but who caused which is unknown. And what the shared genetics is also unknown. So this motivates us to look at this. So we look at curated study from diabetes from different consortiums. Like you can see the sample size pretty large. And for diabetes and resting heart rate based on UK Biobank and Diagram Consortium. We also look at how the traits related to diabetes like fasting glucose, insulin, and BMI, weight to hip ratios, and triglycerides, total cholesterol, and HDL, LDLs, and so on. Right? A bunch of traits that is related to diabetes. And then looking at the overall sharing genetics again, to start with, you can see sharing substantial genetic effect between diabetes and resting heart rate, and fasting insulin, and also fasting glucose. Right. Okay, and some other like TGs and also the significant p-values. Right. So this is interesting. So then we look at the causal effect between them. So you can see for diabetes and resting heart rate, seems both directions are quite significant. Right? The p-value 10 to minus 11, 10 to minus 55. So the causal effect size is related to the unit of the exposure. So we don't directly compare the causal effect size between these two directions because the one is for one yes or no for diabetes, the other is for one beat of the heart rate. But this seems to be consistent to the literature that they do have the two directional effect from resting heart rate control and diabetes. But for others like fasting insulin and fasting glucose, one direction seems to be more prone to the other, right? So for example, it's likely the resting heart rate causing fasting. So this is the fasting glucose causing resting heart rate because this is the outcome, right? The forward means resting heart rate to the glucose. And similarly for the insulin, this is the insulin is the outcome is to be significant. Right? So resting heart rate causing insulin, fasting insulin. Right? And also for others like TGs and BMIs and weight to hip ratio seem to have both direction as well. So quite a bit of sharing between the diabetes and diabetes related traits. Then looking at tissue specific gene expression again, we can identify the sharing of the genes at tissues. Like many of these seem to be expressed in the brain tissue, cortex tissues. This is the one that particularly interests to us that has shared genetic between diabetes and the resting heart rate expressed in the brain cortex tissues. But as you can see, it's also expressed in other tissues. So yeah, looking at several genes. And then we can identify the enrichment of the expression in the different tissues. You can show the enrichment of this. So this is number of significant traits that show up in the different tissues. And compared to the solid line is the number of previous report sharing resting heart rate genes. So by leveraging the expression data set, we can identify more genes that's linking sharing resting heart rate and diabetes. So this is for diabetes. For this is the overlapping for diabetes and resting heart rate. So you can see the jump between the previous report one and the new one. But looking at the fasting insulins, you can see a lot of previous report one already show up here. So the new one much less than the diabetes. So this is for insulin, and for glucose also the gene expression explain a lot the previous identified signal between the resting heart rate. So yeah, so this is for insulin and the glucose. And Okay, so that's the second example for looking at diabetes and resting heart rate. Then I'm going to show some very early study that we look at Alzheimer's and cancer. And also it's motivated by epidemiology study. People find a reverse association. People have Alzheimer are less likely to have cancer. If you have cancer, less likely to have Alzheimer. This might be subject to some survival bias because people have cancer, but they might die earlier. They don't have time to develop Alzheimer. People have time, have the opportunity to be old enough to have Alzheimer, then they might not have a chance to have the cancer because even for some, especially for non-lethal cancers like lung cancers, higher mortality rate. But we try to understand what's the sharing of the biology between Alzheimer and cancer, whether these sharing are actually reversed, has a reverse effect. So this is the motivation behind this study. So again, we look at the summary statistic from the Gammon Consortium. This is some early study published in 2017 for all different kinds of cancers, GWAS, and the Alzheimer. We look at the IGAP Consortium for the stage 1 with the Genoa Association results published that we can combine these two, cancer and Alzheimer's. So as you can see, several of the cancer, especially like breast cancer and prostate cancer and lung cancer, has a shared genetic component between Alzheimer's and the cancers. And they're positively correlated. They're not negatively correlated. So except for the prostate cancer, seem to have some negative effect, but the statistical support are not as significant than the breast cancer and the lung cancer. Okay. So overall, by combining all the cancer together, the overall is a positive and significant genetic sharing between cancer and Alzheimer's disease. And then we can dissect this sharing into several functional catalog in the genome, including what's the introns, exons, super enhancers, repressive regions. So you can see that it seems it's more enriched in the super enhancer region of the genomes. Okay, so this is one way to understand the biology, the controls of this. And then we look at the individual SNPs sharing between Alzheimer's and breast cancer and lung cancer because these are the two types of cancer we find more significant sharing. So we identify these SNPs around these genes to be related to Alzheimer's and these two kinds of cancers. So then we also notice there's a report about sharing of cancer and Alzheimer, but at the gene expression level. But it seems that the effect direction are not consistent with the positive association you find in the genetics. So we try to understand what is the potential mechanism explaining this difference. As we can see in the TWAS, there are several scenarios that the gene expression and the disease can share, right? Either based on reverse causation or causal effect or the shared pyrotropy. And also we can see in the asthma example, the same SNP can have opposite effect direction for the same genes expression in different tissues. So what kind of tissue are measuring in that study will be very key to understand why they are positive correlated or why they are negative correlated. So we put this explanation here to try to understand if you have a positive genetic correlation between cancer, like to Alzheimer, you can have a negative correlation for the genes if you're looking at different tissues. Okay, yeah, because negative to negative is a positive, right? Positive to positive is again a positive. Okay, so we move forward to understand metabolite trait because there was an NIH study showing up the glucose metabolism in the brain associated with Alzheimer's. So we're trying to understand how the genetic control of metabolites or metabolic traits like the glucose or insulins are linking to Alzheimer's. Okay. Of course, we also include the diabetes in this study. So we try to curate a bunch of related traits like obesity traits or lipid traits, including LDL, HDL, total cholesterol, and also the diabetes traits and insulin and glucose into these frameworks. Similar approach applied to metabolic traits and Alzheimer's. And then interestingly, you find that the fasting glucose and fasting insulin has a significant P with the Alzheimer's, especially after we combine them using the meta-analysis, because the insulin and the glucose metabolism are heavily related to each other. So we try to combine the evidence together using the cross-trait meta-analysis. So And also for HDL seems to be also has a shared genetic with the Alzheimer disease. And again, we look at the shared genetic variants with specific traits and the Alzheimer's. Try to highlight that TCF7L2 is one famous gene for diabetes. So this has a significant share of between Alzheimer's and fasting glucose. And we also trying to look at the sharing, whether the genetic variants shared between Alzheimer and the traits are highly expressed in what tissues in the gene expression cortex. We find that like blood, esophagus, heart, livers, and pancreas are in reach of the sharing of the genes. Right. So this helps us to narrow down further to what are the potential functionality for the sharing of the genes. So I have talked a lot about the genetic sharing. Now I want to switch gear a bit to the metabolites as of now. So follow examples. So the motivation of this is from Mediterranean diet intervention study in Spain, that we have 7,000 individuals randomized into 3 groups. 2 groups have Mediterranean diet with high olive oil or high nuts. And then the 1 group is for control diet. I want to see, follow up 5 years, 6 years, whether the Mediterranean diet can prevent heart disease better than the control diet, right? They have all involved without that heart disease, but they can have high risk, like they are obese, they have high blood pressure, they can be diabetes patients. So we want to see whether this healthy dietary can prevent them. They do show that has a healthy effect on preventing heart disease. So we're now trying to understand whether metabolite in the plasma can tell us which one are more closely follow Mediterranean diet, which one are less closer. Even though they're Spanish people, many of them are follow Mediterranean diet, but some there's a large variation about adherence to the diet. So this is how the measurement here. So I'm here. You can also do your own exercise to show. If you answer yes for each of the question, you can get a point 1, and then you add up together. The highest point is 14. The lowest point, of course, is 0. Right? Can you guess how many score they get in the Spanish people on average? It's 12. But the range can be from 8 to 13 or 14. So it's a large range, but they generally have high adherence to Mediterranean diet. So we also measure this LC-max based metabolites at the Broad Institute. So about 300 metabolites. We try to link these 2 together. We try to build a prediction model by using these 300 metabolites to predict the score on the right. Then they tell us whether this can be used to create a signature for the dietary intake. So we leverage the machine learning methods like elastic regressions to do this, because for this analysis, we can regress out the reporting error, right? So the reporting error do not change your metabolite in the blood, but it can be included in your questionnaire answers. You might be recording some healthy food better than the unhealthy food. If you are asked, what do you eat in the past week or past year, and check the frequency of different kind of food and how much you eat, you might be have different recall for this food. But the metabolite in the plasma is more general, more honest to the biologists. And then we apply this to not just the Spanish study, also the US-based study for nurses health study and health professional study in the US, based on similar platform metabolites to verify this is really predictable to the disease. So in the end, we can build a signature of 767 metabolites to measure the Mediterranean diet adherence. In general, the correlation about 0.3, not very perfect because we know there's a response different. But we can see that the signature can tell us whether it can have a risk for CHD and stroke in the next 5 years. The effect size are pretty strong, even stronger than the questionnaire-based assessment. So this is the advantage for building a biological signature for the dietary exposures. And also based on Mendelian randomization analysis, then this is also consistently to show the causal effect, a protective effect. So, so we utilize this strategy to the MindDiet and Alzheimer's disease that we recently published in the Journal of Alzheimer and Dementias. Because for the MindDiet, it's the idea to combine the Mediterranean diet and DASH, which is dietary pattern to stop hypertension, because they take the element that they think to be useful to preserve cognitive function and reduce the risk for dementias and Alzheimer's, to build a combined dietary pattern, so-called the MIND diet. There's an observational study also show that MIND diet can lower the risk for dementias and slower the cognitive decline over the time. So now we want to see how this dietary effect affect the expression in the brain, and whether that can help us to linking to dementia over the year before the death. So we leverage the two study run at the Rush University at Chicago. There's a two cohort. One is called the ROSE, the other called MAP. They are enrolling the individuals in the community, and then they have very structured neuropathological examinations, have a very good score for every year before the death. And they're all free of no dementias, and they will donate the brain and muscle and the serums after death. So overall, there's more than 3,000 participants were enrolled in the ROSE and MAP, and a very high follow-up rate. And we have the also expression data in the brain after the death. Okay, so in terms of cognitive function assessment, there's very detailed tests for the five domains. And then we have the individual score, and then we sum together the overall score. So also, because they are measuring multiple years, so we can measure the personal specific slope for the decline of the cognitive functions. So we want to understand which one are decline faster, the one other decline slower. And they have the dementias definitions, and also some mild cognitive impairment, not severe to be defined as a dementia, but impairment for this phenotype to be used in these studies. And in terms of dietary intake, they have the food frequency questionnaire, like traditional epidemiology study, in one of the cohort, but not the other one, just in the MAP cohorts. We have the MIND diet score. It's 144 food items to ask what they eat, and then they can calculate the MIND diet score. So whether they closely adhere to MIND diet or they loosely follow the MIND diet. Okay, so based on this, we can measure this is the expression data in the brain, in the front cortex front of the brain. And then we also have a subset with 400 individuals with the single nuclear RNA, so that you can see the expression in different subtissues, in different tissues in the brain. Okay, so this is used for the functional analysis. So this is the basic studies characteristics. Like we start with the 400 individual with both the sequence data of RNA and also the MIND diet score, and then we apply the signature to the subset of 700 individual that without the food frequency questionnaire score, but have the gene expression score. And then for both, we can link the RNA expression in the brain to the dementia's outcomes. Right. So this is the overall strategy. You can see this similar to what we see in the Mediterranean diet for heart disease. We first build the signature, which consists of 50 genes expression to be predicting the MIND diet score, the adherence of MIND diet score. And then we apply this to the rest of 700, and then combine to see how this score is linking to dementia's risk. And as you can see, both score, they are measured multiple times over the year for the cognitive functions, and then they donate the brain after the death. This is the overall settings. Okay, so 50 genes, 23 are positive correlated with MIND diet, 27 are negative correlated with MIND diet, and some genes already pop up that has been previously reported to be related to education, attainments, and other cognitive performance in depressions and other studies. So some genes already pop up that linking to MIND diet. And then you can see that no matter the MIND diet score based on FFQ or the gene expression signature, they are positive cognitive with the slope. So having a good dietary score have a better protection on the dementias. Right. So showing up here, not just for the MIND diet, or not just for the mild cognitive impairment, but also for dementias. Right. So dementia seems to have a larger effect size as well. Okay. So based on this expression, we can identify this gene expression signature for gene expression in the brain that do associate with dementia's outcomes. And for the mediation analysis to try to understand which gene expression are responsible for this total effect, the several genes pop up here. The 5 gene mediate 41% of the total effect from the myelin to cognitive decline trajectory. And single nuclear RNA sequence data tell us pop up some tissue like neurons, and these tissues are also maybe responsible more on the association for these genes. Okay. So in the summary, we identify a brain expression signature for the myelin, and we also see that these signatures are associated with the cognitive decline in those patients, and some top genes seem to be quite interesting to follow up with their very interesting pathways. So at this point, I would be very keen to collaborate with basic scientists to go further down to understand the biology of this, maybe based on cellular model, maybe cell type model or animal models to follow up. So finally, I want to outline that our labs in the future, what we are trying to do is to integrating multi-omic study in larger scale longitudinal cohorts for pediatric cohort, for adult cohorts, in the setting for observational study or in terms of intervention trials, like dietary intervention or some clinical trials as well. I think this is quite important, and it's ready the time that has available multi-omic study. In particular, for Alzheimer's disease, we have 3 grants coming in just for the Alzheimer's and brain-related multi-omic studies that we have recently started. So finally, these are all the collaborations that I have been enjoyed with. You can see this is a lot of work by a lot of collaborating natures for different disease areas, different institutes, and different countries. So I like to continue this type of the style of work with people, including people at Hong Kong and university here. I like to explore more collaborations, so including your names here in the future. Okay, finally, this is my labs. As you can see, different student, postdoc, they have different interests in different disease. Because the method we develop are already applied to different disease. You can see they do look at different disease, including kidney function and gout and machine learning methods and like others like even PCOS. And so yeah, this is a very free and collaborative environment. So hopefully you have some time to visit Harvard. I would be very welcome to you. Okay, thank you very much again. Okay. I can take questions.
Okay. So thank you, for the highly inspirational and innovative approaches in understanding the pathology of different diseases. And I can see that it is a very powerful, kind of analysis. Yeah. To me, it is very new, but I would say that I understand the mechanism behind, and I can see a lot of potential applications for many experts in here. All right, I don't know whether they are experts from FSN. Any representative from FSN? We have a food science department, so I like the mind diet and also the death diet. And also the relationship with the cognitive decline, I think is a highly potential kind of research area. All right, now although we do not have a lot of audience, it is not small, but we have a large group of people online. So I think we also take questions both here, those who are physically present and those who are online. All right, and so. I would like to ask the first question to start off the discussion, all right, if you don't mind, and so that you can, try to figure out, what kind of questions we would like to ask Professor Liang. Also, now I'm a little bit disappointed that because you did not elaborate and give example on your method and understanding the pathology of schizophrenia, and which has been my primary area. All right, so I started off the question like that. So 30 years ago, when I was a PhD student, I read a review article on the etiology of schizophrenia. Of course, part of it is on genetics, and I do not fully understand, but a conclusive statement is that there has been no breakthrough, all right, 30 years ago. and they are reviewing so 10 or 20 years even earlier. And I think it is in Archives of Psychiatry. So no, there's no big flu. And recently, I read another review article in World Psychiatry, right? So they are reviewing the same thing, the etiology of schizophrenia. And when I read through the section on genetics, they are making a similar statement. There has been no breakthrough. But I can see that, your method will throw light on understanding more about the brain mechanism and also physiology or pathology of schizophrenia and other major psychosis. So do you have any ideas on that?
Yeah, so unfortunately, the asthma and schizophrenia in the study don't show up in a significant genetic sharing, right? But that's not necessarily prevent other disease and schizophrenia, especially between maybe mental health disorder and neural disorder. So could be have some similarity or distinct between the schizophrenia. So I think if I would go into this direction, I would probably more extensively linking several psychiatric or mental health disorder with schizophrenia, like a sharing of the first start with a heat map of genetic correlation between all different kind of mental health disorders, just to pick up which one are more sharing or which one are less sharing. Yeah, so this would be the strategy. But also I think not just for the psychiatric or mental health disorder, but maybe other type of disease based on the biology of schizophrenia that you hypothesize that could be linked, and then look for those traits or disease. I don't follow the literature for schizophrenia pretty much, but if do you have any ideas like what kind of similar disease or traits that could be related in terms of phenotypic correlation to be started with? Because like in the UK Biobank and others, you can have several thousand of traces available, so you can run the GWAS for all of them. So now it becomes an opportunity to look at, yeah. So this is a general strategy, but also for in terms of the area for intervention or for exposure. So I don't know what. Again, I think we start with observational studies, see like whether dietary habits or lifestyle or even early year family environment and others maybe relate to schizophrenia. And then I would try to build a signature, like metabolomic signature for those exposures and see if that exposure can be linked to schizophrenia. Because the strategy we find from the Mediterranean diet and the mild diet strategy is we find that the molecular signature for the dietary exposure seems to be higher or more effective to predict disease than the FFQ-based assessment. Like I think FFQ-based assessment is not just the one example for diet, but like for physical activity, people usually ask, right, what kind of exercise you do every week and how frequent you do exercise. So I think this kind of assessment is similar to other exposures, other lifestyle exposures, like even for the occupational environment or family environment, all based on questionnaire. So if they can be reflected into the blood or other tissues, then biological signature could be more effective to be a tools to link to the disease. So I would like to continue this role for building the signature for different exposures. So I think people have been looking at this after our publication of the Mediterranean signature. People look at all kind of signature, including all kind of food like coffee and milk and nuts, and now even for signature for mortality, for healthy aging. Like healthy aging is defined as disease-free before 70 years old, or the first timing of the first major disease like cardiovascular disease, diabetes, and cancers. So so it seems this idea works pretty well to develop a molecular signature for exposure, not for the disease, but for the exposure, and then link this exposure to interesting disease downstream. So I would continue use these two strategy.
Thank you very much. So Li Ming, if you are interested in using your method to study more about schizophrenia and also related psychosis, let us know. So I think we have, you know, a good area for collaboration.
Yeah.
So I think, you know, people in this area, they are still not aware of your method because your method is still emerging. And I think your team has played a very important role in you know, trying or playing around with you know, the data for GWAS or what, and in different diseases, just like what you have tried to explain to us. So like you know, I just you know, one more minute for my sharing of experience. So I last month I was in Guangzhou giving a lecture and having a meeting with you know, some basic scientists. They are not mental health expert, but they're asking me whether, you know, there is the relationship between diet, all right, and major psychosis and schizophrenia. And according to my, you know, if any of you have, you know, other information, let me know. I have been following, you know, literature for schizophrenia and psychosis for a long time. But limited is talk about, you know, diet, the relationship between diet and the development of schizophrenia. But, you know, after listening to your talk, and she, you know, that expert is about I think is a biochemistry expert. All right, she does not she's not doing research on schizophrenia or mental health, but she asked me whether there's a relationship between diet and schizophrenia. And at that time, I said limited knowledge is known about this area. But you know, I think you has inspired me that I may be having a different answer when I see her. You know, later on. Next week, I will be seeing her again.
Yeah,
I will be talking about your research to her.
Maybe we can generate some preliminary data before you meet her.
Yeah. That's great. All right. Okay. So that gives enough time for you to warm up your mind and ask questions, right? So I give the next question to those who are physically present first. So anybody would like to ask? Oh, Pearl. And Pearl, you want me to introduce you? You want to introduce yourself? She's our assistant professor, having a very close relationship with you. And I employed her when I was head, and she is an expert in genetics and also mental health. So what is your question?
Thank you very much, Professor Liang, for this very informative and wonderful talk. A lot of information, and I did learn a lot from you. So let me introduce myself. My name is Prof. I'm from the RS department, so same department with Hector. My background is also genetics, but I'm mainly focusing on the mental health, for example, like Alzheimer's disease and dementia. And so I have learned from your presentation that one of your focus of the study is to investigate the cross-link, the overlapping, genetic overlapping between non-mental health disease and mental health disease. So for example, like the Alzheimer's disease and the cancer and asthma and mental health issues. So I just curious that when you start to linking those two different type of disease, non-mental health and mental health disease, how you come up with these ideas? So based on what kind of, you know, strategy to explore those information or whatever, I mean, how you come up with those ideas to close link those type of two disease? Yeah, thank you.
This is a very good question. So my lab is more focused on methodology and apply the methodology. So half and half time developing new methods and half time applying this method to real settings. I have a lot of collaboration with different disease experts. So for example, the asthma and mental health disorder idea was driving from the asthma experts. So we are actually trying to understand the etiology and the pathway for asthma, especially allergy and non-allergy specific asthma. So from there, the asthma is the primary trait of interest. And then starting from there, we look at the literature about phenotypic correlation between asthma and disease. Of course, allergy is one of the top candidates, but also we have our knowledge about non-allergy specific asthma. So that's why obesity and asthma became the second project that we look at. Right? But also we noticed that there's a report in the literature about mental health disorder, especially ADHD, anxiety, and depressions. are having sharing with asthma in the observational study. They have together. They show up in the observational study together. So that's become a motivation about whether they do have sharing of genes between the two diseases, right? Because the sharing of the two diseases can be because they share in the same environment, right? They can also be because they have some shared underlying gene or pathway. So we want to use the genetic to understand this better. So that's how the motivation of asthma and mental health disorder are coming out. So that's one. For the Alzheimer and cancer, it starts from a collaborator at the VA hospitals. She's interested in Alzheimer, and we noticed that there's a famous reverse association between cancer and Alzheimer. But many of them are thinking this may be due to survival bias. People have the cancer, they could be die earlier, they don't have time to develop Alzheimer's. But they also share biology that has hypothesized this. So that's why we start with the genetics to see if they do share genes and whether that's a positive or correlated. It turns out it's pretty complicated. Yeah, overall seem to be positive for lung and breast cancer, and overall seem to be negative for prostate and Alzheimer's. But if you look at individual gene, they can be positive or negative correlated. And if you put also the layer about gene expression regulated by those genes, it can be more complicated because if you look at different tissues. So yeah, so this also can be a direction to the next steps to narrow down to those. Yeah.
Okay, thank you. Now, because I'm still waiting for more specific question. There's one, but not very specific. So that is why. So I think we can continue to take questions from the floor. So anybody? So Philip, please introduce yourself first.
So hello, Professor Liang. Thanks for an interesting talk. I'm Philip Ho, also from RS. So Hector just brought me in. So thanks very much. So my research basically focused on the basic research of Parkinson's disease. So now Parkinson's disease has no cure, and environmental toxicity is one of the key factors that affect. Basically, is a key factors of Parkinson's disease. So I have an ongoing studies looking at the effects of various environmental toxin on neuronal health and how our exposure to environmental toxicities can affect disease progressions, because now there's no cure for the disease. So controlling the disease progression is also one of the key focus of the research in this area. So my question is if there's any way or maybe your opinion on how to apply your cross-trait analysis to link these, to look at how environmental toxicity can affect disease progressions. Because the reason I ask, because we have some pilot studies looking at various environmental toxin, and we have recruited a cohort of Parkinson's disease, and we have collected the plasma, and we found these toxins are readily detectable in the plasma. So, but there's still no any comprehensive studies looking at the toxicity of these toxins. So I'm trying to see if exposure, a long-term exposure of these environmental pollutants or chemicals, how can it, using this method to correlate with clinical scores or the outcomes. So
yeah. So environmental toxicity, basically environmental exposure, source could be less genetics, right? It's a non-genetic, so it's exposure. But for the people that are having the same exposure, they may have different ability to metabolize or remove this kind of toxicity, whether the biology allow them to be letting them out from the urine or from other body fluid. So if they do have heritability, so I would look for first whether evidence of this environmental toxicity of like even the heavy metals or other toxic components have a heritability. If they do, then you can link them with Parkinson's disease using the genetic approaches. So that's one immediate idea. But For this application, my thought was maybe the second method would be more appropriate. Like, and you know, there are very hot topic right now, so-called exposome. Exposome is like one kind of omic, like metabolomic. We are measuring all kinds of metabolites, small molecules in the blood or urine. But exposome is to measure all kinds of exposure, like environmental toxicity, one of them in the urine, and to see how the individual are being exposed to different environmental toxins in totals. So many of these techniques are based on mass spectrum technique to measure, very similar to metabolites. So you can treat them as, in terms of analysis, you can treat them as metabolomic studies, but you can also build a signature, like exposome signature for certain kind of environmental toxins, and then link to Parkinson's disease or other neurodegenerative disease. I think this approach might be working even better than the genetic approaches, because it really measures the environmental exposure. This is really the source. And I think there are also available technique, as long as you have the plasmas or the biofluid like urine in a cohort study, then you can start with that. Yeah, the mass spec profile, the exposomes. Yeah. So this would be one of my new area to work on. Because several my long-term collaborator, Angela Baccarani, was in Colombia, and now he's back to but now he's back to the school as a dean. So he's interested in all kinds of environment exposure. So exposome is one of the things that we talk a lot very frequently recently. So yeah, if you really go on this direction, we should talk more about this.
You said that we might have chance to collaborate to validate your findings, because I'm focusing on the basic science and use mouse model cell cultures, and we develop various different types of assay looking at subcellular activities that fits well to what you said. You just said that to validate your findings from the genetic analysis.
Yeah.
Thank you.
So. Huh? Oh no. Yeah.
Very inspiration talk, and thanks for your sharing. My name is Zoe. I'm a PhD student of Professor Hector Zeng. And I am working. I recently conducted studies on predict factors, heart rate variability, and on depression symptoms. And during the research findings on the cardio-related disease and depression, I found that people with depression symptoms have a higher risk of the cardio-related disease, and cardio-related disease have more potential to get depression symptoms. And under that situations, they could share a genetic related on the inflammation factors or serotonin pathway. And I was wondering if that's the genetic method you mentioned could used in our studies, just on the interventions, investigation studies, or other cross-sectional studies to be a predictors for the depressions, and also about the heart disease, because the heart disease index is not reliable when patients with cardio-related disease, but it's reliable on patients that only have depression disease. I wonder if this kind of index, are they have reliability to predict both depression and cardio, combined with your genetic method? How can we achieve such kind of predictions? Thank you.
So yeah, maybe I would learn from you first. And you mean the cardiac is more accurately measured in the depressive people with depressive symptoms, but less accurately measured in a more general population, right? So if that's the case, then a more refined genetic effect from the cardiac would be built based on the genetic association study in the people in a population with depressive symptoms first. Like you can leverage the study like UK Biobank and others, that if you have enough number of samples with depressive symptoms, then you run the genetic effect in that sub-cohort to have a more accurate estimate for the genetic effect on the cardiac disease. Yeah, if that's the goal, to build a more reliable genetic prediction model for cardiac disease. Is that the goal? That you want to build a more reliable prediction model for cardiac disease?
It's not only for the cardiac disease, but also for the depression. I want to say if one of the heart-related index have the potential to predict both the depression and the cardio and about the accuracy.
Yeah. So I want to point you to one of the methods published in Nature Communication in 2018. It's the so-called cross-trait prediction models. So that is a method to leverage the genetic effect from different traits, then to build the genetic effect in the secondary trait into the primary trait of interest, because they do share genetic effect. So if you incorporate genetic effect in other trait, you basically increase your effective sample size of the primary traits. And sample size is the key for building a more accurate genetic prediction models. So in that method would help you, say, if you are interested in predicting the depressive symptoms, but you know cardiac GWAS can have a shared genetic effect. So building cardiac as a secondary trait and depressive symptom as a primary trait, you can increase your power to predict the depressive symptom as a trait. Yeah. And at the same time, you can take a range of different disease as a secondary trait, not just the cardiac, but others that has shared genetic with depressive symptoms. So on the other hand, you can treat cardiac as a primary trait and depressive symptom as a secondary trait, and maybe others traits as well to boost the prediction power for the cardiac.
Okay, and we also thank you for your answer. And we also doing the intervention experimental. So I wondering that if our intervention could change some expression of kind of short-term gene expression, does it mean that we could be a more stable potential indexed for the how to say the recovery of the depression and also depression symptoms?
Yeah, so depend on how do you collect the data. If do you already have the data, or you will collecting new data? Maybe.
I already have some data, but data set is small.
So the data is collected at different time point before and after the intervention?
Yes, before and after intervention.
Okay, and you can see how, maybe you can talk to the detail later on, but I think one thing to pay attention is if you do have, say, in the intervention settings, you have the expression measure before and after the intervention, and then you should also have the efficacy of the intervention on the, say, depressive symptoms and others, right? So from that setting, you can build signature or expression profile that linking to the effect of the intervention, and whether that effect would be linking to the disease depressive symptom outcome. Because the individual assigned to the intervention arm and the control arm, they might adhere to the intervention differently. So the expression profile can tell you which one are more closely follow the intervention protocol and which one are lessly follow the protocol. The one that are more follow the protocol maybe have an effect size larger than the other one. But if you just look at the intervention arm compared to the control arm, you might have less power to see the intervention effect. So that's similar to the MIND-DIE interventions. Even the MIND-DIE is not, the Rose and Map study is not intervention studies, but the signature in the brain can tell you the adherence. And in the Pradimer study for the Mediterranean diet for preventing heart disease, that do shows us that the metabolomic signature can tell you which one are more closely adherent or respond to the intervention than the others. Yeah, so I think that would work. Yeah.
Thank you so much
Sure.
for answering.
We still have 10 minutes, so I think we should at least answer one question online. And this is the question, very generic, and is that will the MIND diet okay in Hong Kong? So I guess it's that asking whether we should be promoting this kind of diet or whether this kind of diet is is common in Hong Kong or what? So it depends on how you would like to answer this question.
Will the mind die okay in Hong Kong? So it means whether mind die can be applicable to people in Hong Kong. Yeah, I think that's absolutely the case. The mind die by design is a combination of the Mediterranean die and the DASH. DASH is a dye that is designed to be preventing hypertension, reduce the blood pressures. So basically, a lot less sodium to use is one key factors. So Mediterranean diet is characterized by having more vegetable, more fish, less red meat, and maybe a cup of wine sometimes, and olive oil, of course, and yeah. So I think those are more healthy diet. People have learned this in different newspaper and literatures. So it should be absolutely okay, and you can find that a lot of restaurants, they are selling salad bars and many others in healthy. So yeah, so I think that absolutely be applicable, but just be in terms of public health, like the government or the school of that the university want to promote more healthy diet, then maybe give a more interpretable guideline for the public to follow. Like in Harvard, we have a so-called a healthy eating plate. We draw a circle, and then to show what portion of your plate, if you eat the whole thing in your meal in today, like lunch or dinner, what portion, like more than a quarter should be vegetable, and less than a quarter should be healthy protein, like less fat and more protein, like an egg and whole grain, and for carbohydrate, and yeah, and food in the last quarters. So, and more exercise as well. So yeah, so I think this kind of product we translate that into 24 language on the world and post it on the Harvard website. So if the university or the department want to promote healthy diet for mental health disorder preventing, then you can have some kind of advertisement or information web page to the public, and then to educate the people how to follow the mind diet or other Mediterranean diet better.
So I think it is very appealing to people like us in the university, because we are afraid of Alzheimer's disease. So I think promoting this kind of mind diet, I think should be okay within the university. I observed that in Hong Kong it is not very popular, but quite popular among the professional and also the educated community. So I think there's room for us to further promote it. All right, okay, so let's come back. We still have a few minutes. Maybe we can take one or two more questions. So Aaron. Please introduce yourself.
Hello, Professor Liang. Thanks a lot for your insightful talk. I'm Erin. I'm an RAP in RS, and Hector, Professor Zhang is my supervisor, so I follow his research interest in studying Qigong, a kind of mind-body exercise, right? And in our research, we always try to look for the biomarkers that can help us to understand the mechanism underlying the treatment effect of Qigong, because we found it effective to alleviate depressive symptoms, but we really don't understand why. So we are doing it in the conventional way. We are looking for changes in single biomarkers, for example, salivary cortisol. And your multi-omic approach is something very revolutionary. So I'm wondering if this approach can, change our way to understand the potential mechanisms underlying this kind of intervention. Yeah, thank you.
Yeah, definitely. Yeah, I know that. Yeah, for sure. Yeah, I would be very curious to see what's the biological impact for exercising Qigong. We have some relevant experience about meditations and how meditation relate to, say, the brain wave and also the plasma biomarkers. So I think this is something that I would be very keen to see. If you do collect biomarker in the plasma or urine of the individual, measure their metabolite, and see how Qigong exercise can be affecting to those signatures. I think you probably need a way to measure which one exercise more than the others, who is the better Kung Fu master, who is can practice more. So I don't know how do you measure who knows Qigong better than the others or who exercise more than others. Can you have a objective way to measure that?
Yeah, this is actually linked to my next question, because in our way, our intervention is just like we have this 13 weeks of training courses, and we are looking for whether you attend all of them or just attend like 80% of them. So we only have that kind of attendance rate as a kind of adherence indicator. Yeah, we should really go further to measure their self-practice, duration, frequency, etc. So yeah, we don't really have that behavioral data at the moment, but this is definitely something we will do. Yeah, and then my next question is actually about the signature that you mentioned. So that kind of genetic signature, is it something specific to the sample that we have, or it is something that's like general? So if we identify the signature, we can use it as a kind of assessment for our future study.
So taking the example for the Mediterranean diet that first developed based on the sample in the SPAN intervention trial. And then we apply that signature to a US-based cohort. And we do see a similar association with the signature and the FFQ, the questionnaire-based assessment for dietary intake. Very similar correlation. And the effect for stroke and CHD are also similar. So it's applicable. So the idea for this method is that people have a high score of signature, means their metabolite profile are very similar to the people who have high adherence to Mediterranean diet. So if you were trying to build a signature for this Qigong exercise, and you only have the assessment about attendance, although attendance is not a perfect way to measure how they exercise Qigong closely or loosely, but if your sample size is large enough, you can still tell people have high attendance. In general, you would reasonably believe that they have a higher quality of the Qigong exercise than people with low attendance, right? And then if you can build a signature, then the higher score of the signature means the individual, the profile in their plasma will be similar to the individual that has a high attendance of the Qigong, right? The individual getting high score, they may be because it's really have a high attendance for Qigong, or it responds better, even though he exercised less, but his genetic background and other part, maybe the guy is already in the mind of Qigong all the time, even though he don't attend Qigong exercise section than others, but he's reflecting in the plasma already. So that's the in terms of food, it's about intake and response. In terms of Qigong, it's about exercising and response. So the total effect is similar to individual with high attendance. So you should expect that this high score should be related to the good results for Qigong if Qigong has a good results, right? So I think this strategy would work. But of course, I would be very put in the effort about measuring how to measure the quality of Qigong exercise, not just by attendance rate, but also maybe based on some post assessments and how they feel and really, I don't know what's a marker that you can use, but I believe there would be some markers. And then you use that score for your training models, the outcome of the training models. Yeah.
Thank you very much.
Very interesting.
So I think this is the last question. So the gentleman at the back.
Hello, Professor Liang. Thank you so much for your presentation. I have learned a lot. And my name is Frank from RS. My supervisor is Arnold Wan. And I'm looking on the chronic low back pain right now and using fMRI or structural MRI to look at their brain mechanism. So I also noticed UK Biobank have some brain imaging and have the gene data like the GWAS of the brain imaging. Yeah, because they have just put out the data. And UK Biobank have opened up a IIP research platform. So I just wonder if you have any experience that can share with us about how to make full use of the brain image data. Thank you so much.
Yeah, UK Biobank had the brain MRI and also different part of MRI available. We actually run the GWAS on each of these MRI traits. I don't have the time to present here. that was one of the study we link the stroke and Alzheimer together, and also the brain and also the breakthrough metabolite GWAS as well. So yeah, we do have those available. And if you have access to the RAP, the research analysis platform in UK Biobank, you can run the GWAS by yourself. It's pretty straightforward. Yeah, it's a good resource to link to other brain traits as well if you're interested in other parts that's related to brain MRIs. Yeah.
Okay,
And we
thank you
do
so-
see quite substantial genetic heritability of the brain MRI. So I think there's a good opportunity. Yeah.
Yeah. Because seldom article have not published about using the brain image GWAS data. So I just wonder if you have any experience to run data before.
Yeah, we have those data available.
Okay,
Yeah.
thank you so much.
Yeah. We can, if you're interested, we can collaborate on utilizing that. We link already those with the stroke GWAS, but if there are other disease of interest, then we can link to others. Yeah. But the data is already there. We have get the summary statistic for this brain MRI GWAS for each of the trait. It's pretty strong power for genetics effect over there.
Yeah, that's true. If we have any possibility that can learn from you.
Sure. Yeah. Feel free to email me to follow up any
Thank you.
interest for collaborations and sparkling new ideas.
Thank you so much.
Answer. So Sylvia, you like to say a few words? Oh yes, you want to do it? You want to do it? Yeah. All right. So now before we do it, just another round of applause to show appreciation to our speaker.
Thank you so much.
I must say, this is not something routine, but the word inspirational, innovative, impressive, I think all are, genuine and deep in my heart. It's really like that. All right. Okay. So I think we have concluded this seminar.